Today is Wednesday, February 3rd, 2021. Our guest today is Rex Ma. Welcome. Rex is currently a PhD candidate at the University of Toronto as well as the Vector Institute for Artificial Intelligence. He's currently doing research in the Goldenberg Lab as well as the Wang Lab. His field of research is in machine learning, artificial intelligence, and healthcare, and computational biology. Rex, welcome to the show.
Hi, hello. I'm very excited to be in the show, Gary.
Great. We're super excited to have you here today. So, how are you doing? How are you doing? What's going on? What's going on in your life?
It's going well. I think I have a very busy day today. Lots of stuff,
What do you
and
do?
I did some research. I also did some startup work, like the side project stuff, and
Mm.
also prepare for your podcast. Went through the questions and then do some minor research. Yeah,
Oh
so it's
man,
fun.
it's an
Yeah,
honor to have you prepare for this conversation.
my honor, my honor.
So why don't we dive right into that? So you said you
Sure.
do research. It's very interesting. So you're at the intersection of healthcare and computer science. Such as the term computational biology. Maybe not healthcare in specific, but can you tell us more about like what is computational biology and its like broader meaning? Like what does it do?
Yeah, for sure. So for computation biology, it's the science of using biology data to develop algorithms or models to understand biological systems and their relationship. So basically, it's like applying the current either machine learning or statistical methods to biological data, and then try
Oh,
to learn from it to understand the mechanisms and find out the relationship.
what kind of biological data are we talking about?
Yeah, that's a very good question. It's actually very broad. So one can be genomics data, and then neuron
Mm,
science, neuron data, and also some kind of
wow.
other stuff that, for example, like protein stuff. So there's so many other subfield that I don't even have much knowledge about, but there are tons. Yeah.
So this AI, which has been booming for like the last number of years, at least
Yeah.
like 5+ years now, is now being applied on genomics data, like your
Mm-hmm.
DNA, your
Mm-hmm.
neural signals, like your EEG data. I'm guessing, as well as like all these
Yeah.
other biosignals that can be collected. That's crazy. So what's like the goal of this? Why are we trying to predict something? Are we trying to like diagnose someone with cancer using all of this, or what's the purpose of all
Mm,
this?
I see. So I'm more familiar with the computational genomics, so I
Okay.
can share some of my knowledge on there. So for example, have you heard of like the two most important like the progress in biology in the 21st century?
Is it the gene editing CRISPR Cas9 protein? Is it that one?
That's right. That's one of them, and the other one is the single cell data. So
Single cell data. So
let's
what is single cell data?
yeah, let's start with the first one though. So like
Okay.
maybe everyone knows the CRISPR thing. So the CRISPR, it's gene editing tool,
Mm-hmm.
and it basically just like you can apply that to edit the genes for a um live
Live cell,
organs, live cell, live
which
everything.
is crazy, which
Yeah,
has
and
previously never been done before. It's like,
exactly. So you can create the new species and probably can create lots of cure for the disease. So lots of potential apply using CRISP. But I don't have much experience working with CRISP, so I cannot talk more about that.
okay.
But for the other one, like the single cell data, I do have some experience on that. So it's actually very, very cool. So for single cell technology, it's previously we can only analyze the cells in the bulk, like in the bulk of cell. Like
Okay.
our technology is not matured enough to separate each one of the single cell and then to get their data for each single cell.
Okay, I see what you mean.
So yeah, so maybe a good analogy is, let's say you like fruits, right?
Yeah.
And then like the bulk cell analysis is like a smoothie. You mix everything together, and you can only do the analysis like a mix of everything,
Right, and you
right?
don't get
In
like
that
specific
sense,
information.
exactly. And right now we can isolate every single cell in the analogy way is we can get a one apple or one orange and one grapes. Like you can just pick up any one of the fruits and then have have all the things you like
Okay.
in that sense. Yeah.
Okay.
So
Why is this a big deal? Can you what like before why like it why is this important to like understanding
right,
biology?
right. First of all, I would say by having the single cell technology, we have the ability to look in the more subtle cell subtype that previously cannot be identified.
Oh, like like different like even if it's the same organ and the same like same cell, it's there's
Yeah,
different subtypes of cells.
exactly,
If it's like a
exactly.
heart muscle cell, it's like there's still different types of heart muscle cells.
That's true. And also, a very direct application is for cancer research. So like for cancers, our gene is mutated, and can develop any possible subtype, right?
Right.
So if the previous bulk cell analysis, because all the expressions are averaged, so we might not be able to identify a specific mutations in one of the subtype. But now with the single cell technology, we can.
Oh, okay.
And yeah, and
So,
also another very cool stuff applying single cell is we can identify the trajectory of the cell's evolutions. So
oh,
basically, we can collect the data of let's say one type of cell at multiple time step. So because the cell is keep developing and then can turn into different other type. So for example, every cell starting from the blood the bone marrow cell, if I remember right.
okay.
And then the bone marrow cell can finally like group to all kinds of different cells, right?
Yeah,
So
okay.
with the single cell technology, we can track the trend, like the whole trajectory of how the cell from one type become the other type. That's super cool.
Oh, okay. So basically, before this was all impossible because like when we did try to do any sort of analyses on cells and kind of it was always like a macroscopic mixed picture of
Mm,
what's going on, and now we
mm,
have the single cell like what is it called like analysis method like single cell method,
yeah,
and now we can basically see like one by one and more accurately track what's going on because like a cell is a basic building block of life, so
exactly.
we are able to understand like the full progression. So what are the implications of this then, because this like I am still struggling to understand like after we know all of this, is it gonna like is it gonna solve cancer, like what like that's probably jumping the gun, but like what can happen, like what are the problems that it's able to solve?
Mm, so right now, I think the single cell technology is can be applied to everywhere.
Okay.
So, for example, if we wanna, for example, like cancer cure for sure, right? So, let's say we can identify a different subtype of mutation of the subcells. So, with the cell data, we can do better like a drug development on that, right? So we can
Oh,
identify the targets for that specific cell type, and then we can identify which type of drug can cure that kind of cell, right?
oh, that makes a lot
And
of sense.
other than cancers, for example, like the agricultures. So if we want to plant some new species um, that can live or grow in a very different environments, we can also use single cell technology to identify like a set of cells that can more adapted to that environment, right? So like yeah, so lots of potentials, and then basically you can apply it everywhere. Right
Right.
now, even for like the patients, some have the gene identifications, like the gene test.
Okay.
I would believe that they will switch to a single cell scope for better
Oh,
accuracies, I guess.
right. This is fascinating. So, wow. So, and like because this is cells applies to every organism, we can basically run this analysis on any
Mm,
human being, any cell within the human body, and learn
mm,
from like the single cell technology of what's going on. So,
yes.
what's gonna like is it what's gonna be needed for us to like use this, like is this Is this technology like available now? Is this currently under development? Is this future talk? Like when can we see this happen and like affect our daily lives?
Well, actually, like the technology itself is pretty mature now, but I think okay, so like for single cell technology, they are the technology to isolate into every single cell and then look into each cell to collect the data from there. That technology is very mature right now.
Mm.
So for example, we can collect the DNA of the cells, RNA expressions, their mutations, informations, also some like the protein informations. They are all related, like all looking to the single molecule level.
So
So
if I understand this right, so let's say hypothetically we have a certain cancer patient, and we were able to extract let's say this patient has a cancer like a skin cancer, right? Then we can hypothetically get a skin cell from a healthy part of their body that isn't mutation, and then we can also sample the cell from cancer cells, and then using the single cell technology we can read the DNA sequence, the RNA sequence, all this other information, and then like pinpoint what like mutation has gone wrong to like create like drugs or treatments and otherwise to help these cancer patients.
yeah, that's true.
Wow. So
So
this is a game changer.
yeah, and I can talk about the data type that can generate from single cell technology. So
Okay,
as
okay.
so as I mentioned, so we can have DNA, RNA, proteins, mutation, all sorts of stuff, right? But basically, they are just a giant matrix. So with for example, like the column as the different cells,
Okay.
and the rows will be like their genes or their corresponding proteins, and
Okay.
the like each entry will just be the expression value. So it's just a giant matrix.
Like a giant
And then
matrix of numbers
yeah,
or like
yes,
letters. What are
they are just numbers. So for
okay.
example, like in cell A, gene A is expressed by 2 times, and then gene B is expressed by 5 times. So
Oh.
just a giant matrix,
So
right?
it's not right. That makes a lot of sense. It's
Mm-hmm.
like, like we learned that, DNA genes are like ATCG molecules that are like bind in this alpha helix structure, but it's really like these sequences of genes that characterize like what is it, like gene expression, you said?
Mm-hmm.
And then we see if this how do you measure that, like what's like where does it go from these long strands of ATCG to these like expression
These
A and
numbers.
expression B and like numbers, like well, how do you measure that, like how does that
Okay, okay, that doesn't fall into my expertise to be
okay
honest, because that's really from the
the wet
like the
lab.
upstream. Yeah, it's from the web lab. It's like how they use the giant machines to do some centrifuge, and then put each one of the like the cell into one slot, something like that. Yeah, very fancy stuff there, but I'm not too sure to be
Oh my
honest.
goodness, this
Yeah,
is still
so
so fascinating.
like for ML researcher or the computational biology researchers, we only starting from after we getting the matrix.
Mm,
Yeah,
okay.
and
So you have this giant matrix. What can you like? So what's kind of like the main themes of research these days in computational biology? Like after you have all this single cell data, like what are you trying to predict? What are you trying to like find patterns
mm,
of? Like what's the main kind of learning that you want to extract out of this like single cell knowledge?
right. Um, I think there are the two currents, or like kind of a very mature subfield. One
Okay,
is doing the cell type clustering.
cell
The other
type
one is
clustering,
the other one is as I mentioned is the trajectory analysis,
trajectory
and also
analysis,
yeah, and also some differential gene analysis. So I will go
differential gene
into each one of
analysis.
them. So for
This
example,
is
like the first one,
fascinating. So the first one, yeah, okay, cell
yeah,
type
like the
clustering.
cell type clustering. So that one, it's more easy to understand, I would say. So let's say, so you have a giant matrix, right?
Mm-hmm.
But definitely some of the cells are within the same cell type, and
Right.
some of the cells are from another cell type.
So
So
quickly before you say anything more,
yeah,
like what's an example of a cell type? Like let's say I have skin cells. Like is it like face skin type versus like arm skin type? Is it that? Or probably not, right?
Okay. Probably the only example I can give is the white blood cell and red blood cell.
Oh, I see.
Yeah, for example, like that level.
Oh, I see.
Yeah,
Okay.
and also some kind of like the like microphage. I think that's also another cell in our immune system.
Right.
Yeah, so that's by cell type.
Oh, I see what you mean. Okay.
Yeah,
So
and
one the step the like you basically from all this information without like knowing what it is, you basically can classify these individual cells into their categories,
yes,
clusters.
yes, that's right, that's right. So that's one applications or one research topic,
Okay.
but that field it's well studied, to be honest.
Right.
Lots of methods, lots of algorithms for doing that. And the other one is trajectory inference. I think 1 or 2 years ago, that that one is still a very hot research topic. Right now, it's more mature right now. So
That's
for
fast.
trajectory
That's a
inference,
fast.
very, very fast. So
Right.
single cell method development is going crazy right now.
Mm.
And for trajectory, it just like as I mentioned, because you have different cell type, right?
Right.
But different cell type are developed in the time series fractions. So
Okay.
within one time point, um, one cell type might be the parents of the other cell type. So they are in a development process, right? So how you will getting their relationship just from the numbers.
Whoa, this
It's
is
also a really good
fascinating.
question.
So,
Yeah.
like a cell can reproduce itself, right?
Mm-hmm.
But in your and these cells can like after they reproduce, like a new or split off into a new cell, they can be split off in a different cell type.
Yeah.
Wow.
Exactly.
Okay.
And
Definitely
by
didn't learn this in biology class when I was in high school. They told me it was the same cell. Now I know they lied. Well, at least they didn't know.
yeah, exactly. So how fascinating it is by just looking at the numbers, right? Like gene expression numbers, then you will know like, okay, this cell is coming from that cell type, and this cell is going to that cell type. So that's yeah, it's very crazy.
Wow, so you can basically, if you have this sequence modeling, then you can almost infer if this one person will have a particular illness in the future or not, or will be healthy or not. Is that too far, or is it
That's too
possible?
far. Yeah, I would say um
Like
so
if my family has heart problems, right? Let's say, or like
mhm,
someone's family has heart problems, and it's caused by this degeneration of the heart muscle cells, like becoming weaker
mhm,
and weaker. Then
mhm.
can we understand earlier on that like we sampled all these like heart cells it's going bad. Is that possible to information to get?
If it is a genetic disease, like defects in the very beginning, probably we can do that.
Okay.
But for example, like if something goes wrong, for example, mutations happens, that thing is not controlled by ourselves, right?
Right. Okay.
Yeah, so we cannot like that one is very hard to predict.
Mm.
So
Is it
I would
possible though?
say
Is it possible?
I would say that's okay. I'm not too sure, but I think there's potential in there.
Mm.
So yeah, so right now trajectory inference, there's methods coming out, but as for how to apply them into solving the real biology questions,
Mm-hmm.
it's still a miss right now, I would say.
Okay. Maybe
Hmm.
maybe we can meet back in some time and we can see what kind of problems we can
Yeah,
solve after.
exactly.
So this is fascinating. What was and your third
Oh, the third one, it's simpler. It's called differential gene analysis.
okay.
So, for example, um, so different cell type can have different type of gene that are expressed more, right? So, for example, one cell type, their marker genes, they like the so-called marker gene is like they express like significantly higher than the same gene in other cell type.
Okay.
So how would you identify that kind of genes in that specific cell type? It's called differential gene analysis.
Wait, sorry. That totally blew over my head. So you have the same cell type and then
Different cell
a
type.
different cell type and one of the cells have a higher expression or lower expression in like a certain gene like
Hmm.
expression.
Hmm.
And what are we trying to do with that?
So we can identify which cell type like the marker gene for each cell type.
What is a marker gene?
So let's say cell type A, so like remember the giant matrix I have
Yeah,
told you?
yeah, yeah,
So like a giant matrix consists of lots of cells, right?
yep,
And the cells come from different cell type.
yep.
And the genes are probably just like 20K genes. And so for sure, like let's say for example, like one cell type, they might have only like 100 genes that express um, much higher than the other type of the like other cell type.
And what do you mean by express here? Is it like,
Okay. So do you remember the number I just mentioned?
okay,
So for example, like the gene A in cell type expressed by 10 times, that express is by for example, like let's say the protein thing.
right.
So like the by 10 time is like from that genes we create 10 proteins. We replicate that DNA 10,
Oh,
10 times more than other genes in that cells.
oh, I see what you mean. So
Yeah,
that specific like subsequence of genetic information is playing a larger role in protein
exactly.
synthesis of the cell, and
Exactly.
therefore having like whatever role it is, protein synthesis and causing downstream
Mm-hmm.
influence in the body, whatnot organ, is having a
Mm-hmm.
bigger impact.
Exactly.
Oh, I
Exactly.
see what you
So
mean. This is
yeah,
fascinating.
yeah. So basically that type of questions is trying to type of research is trying to address this kind of questions and
Right.
just identify the marker genes for each cell type.
Oh, so you're basically like figuring out what the job title of each cell is in this
Exactly.
giant matrix of information. That is fascinating.
Yeah.
This is really cool. So I wanna also quickly talk about CRISPR Cas9. If you have information about that to share as well.
Mm-hmm.
What is so like CRISPR Cas9? It promises gene editing of live cells, which is honestly mind blowing. And this technology was like, was it 2014, 2015 now? It's been a while. It's already 2021. It's been a while.
Mm-hmm.
Um,
Mm-hmm.
how much does computational biology play in this specific like role or this technology?
Um, Very good questions, and I can only guess to be honest.
Okay.
So for example, after we editing the genes, and the genes started to replicate, right?
Yeah.
And then it would just like transfer into RNA and then to DNA,
Yeah.
uh, to RNA and then to protein, right?
Right.
So in that case, we want to validate whether the gene we have inserted into that got expressed. So
Oh, okay.
yeah, so like I
Right. You
would say
use that.
computation.
Oh, this is so cool. You can use this technology in conjunction.
Exactly, exactly. So basically for computation meaning, it's just like compute like those numbers, like those matrix to get useful information from there, to do some validations, to prove something. That's the whole thing, just like computation biology, right?
Wow, this is fascinating. I feel like I feel like we're already entering this future. Like now, I'm very curious. Now we talked about some of these technical details. I want to take a step back and see where this when did all of this happen? Why is it like we never heard about gene editing, computational biology, genomics research in the news or anything like that. At least not on mass media.
Mm,
Why do you think there's such a like kind of like an insurrection going on these days? Like why is there more interest these days, and where is this like kind of research headed in the direction? Like why genomics and why now?
I see. I think one of the thresholds is definitely the like the very upstreams of technology. So like for example, the single cell technology is one of the examples. So previous we don't have that type of technology, right? So like the previous analysis probably just like not very accurate on each single cell type, something like that. So with the so it's like a deep learning thing, right? So now we have the computer visions. That's only because Geoffrey Hinton get the deep learning out, right? But with the
Mm.
technology in the very like the base technology is happening, and then now we can do all sort of create more tools on the new data created.
There's a
So
breakthrough in the upstream, and now
yeah,
we can do so many different things.
in the downstream, that's true.
That's pretty
And
cool.
And also, like so in my perspective, I think the healthcare and also like it will always be a very important topics among humans, right? So like
100%
for
agree
example,
with that.
yeah, 100%. So like back in like 2000 years ago, like the China's empire, they want to live longer, right?
Oh,
And
right, right.
yeah, they don't
Yeah.
want to die. They try to
They want
search those kind of
eternal
drugs.
life.
Yeah, so like that things happens like 2000 years ago. So now people also care about age. They don't want to get old, right? They don't want to have
Not only do people
uncurable
want to look
disease.
young, they want to feel young, they want to be
Exactly.
young. Now it seems like it's possible.
It's true. So like, and also, I also look into the ARK ETF.
Oh,
One
yep.
of their biggest, yeah. So one of their ETF is on ARKG. It's also focused on genomics,
The
right?
Genomic Evolution ETF.
Yeah, exactly. So like the lots of focus is on the gene study right now, like genomic study right now. So I would say, okay, I read a kind of saying very long time ago.
Mm.
It's like um, we can solve the aging problems in 30 years.
Whoa, whoa, whoa. Okay, you gotta unpack this. So aging problem,
Yeah.
meaning. So I also, this reminds me because there was this one Harvard professor who says aging is a disease. Like death by age is a disease and it could be cured.
Mm,
So, like there's ways to, there's research about how, your DNA is tightly bound in these chromosomes, have
mm,
telomeres at the end, and these telomeres get shorter and shorter and shorter. And there's also
mm.
other like research about like how aging, like, can you really unpack for me like what does it mean to cure aging? Like aging, like, do you first of all believe aging is a disease, like it's a curable thing?
I don't think it's a disease, to be honest. So like having eternal life is not it's not always a good thing, right?
Okay.
Like
Some
but
people might disagree.
yeah.
Especially those
Yeah,
folks in
but that's
China
more
about 2000 years ago.
that's more physiology thing.
Okay.
But so the mechanism of aging, um, I believe it just like it's so there's something called cell programmable death. It's like it's controlled by the genes.
Right.
So after a certain of times, like the genes will make you defunctions, like for the cell to defunctions.
Right.
So so in that sense, if we can identify which part or which genes can actually do the aging stuff, then we change that with the CRISP, right?
Yep.
So it's possible to not aging, I would say.
Right, because okay, I like, please correct me if I'm wrong. For the folks that like require a little bit more background on this, so basically, evolutionarily speaking, for some reasons, I think some literature suggests that people age and die such that, hopefully, what is the better genes of better genes of their predecessors will live on,
Mm,
and I don't need to hog down the resources. So if I, as hopefully the person with the inferior gene sequences, die off, and my children have the better gene, hopefully they will live longer, and I don't have to waste their time and resources, and I can die off. I don't know. Okay, obviously that's a long stretch argument, but basically the point is, our genes are programmed for us to die eventually. So
mm,
it's hard coded in all of our DNA that we die one day, which is it's scary, or it's relieving, or something. But basically, if now we're saying we can change that particular gene sequence in our DNA in our live cells, and we can essentially live forever or not even age at like after a certain point in time.
mm.
Can we
I
reverse
would
that?
like what do you mean by reverse?
Can we get younger? I obviously won't get shorter. I hope not.
Um, okay, I would say that's a next level question.
Next level. Okay. All right.
That's too next level.
But we can
Yeah.
definitely like at least slow it down or reduce the like expression of that gene, like try to lower that number in the matrix.
Yeah, exactly. And do you know
Wow.
like the cancer cell, they never die because their gene got mutated.
Right.
And then yeah, so cancer cell is one of the example, right? That can live very, very, very long time.
So single cell technology can identify which gene sequence is triggering this problem, and then we can use CRISPR and Cas9 technology to fix those cells, and then we can use single cell technology again to verify that these cells are no longer cancer cells.
Hmm.
This comes full
But
circle.
wait,
This sounds
wait,
like this sounds like we
some,
solved all the problems we need to solve.
wait, wait, some caveat on this one.
Okay.
I'm
Don't
just
give
like
me the
thinking.
caveat. Don't give me the disclaimer. I was having such a great time.
Okay, so like I'm just like thinking very ambitiously
Okay.
to like linking all the technologies. I just like thinking of their potentials, right? But whether they can achieve that or whether it's actually doing this way, it's very hard to prove right now. So we can
Right.
hope.
It's
Yeah,
still work
we
in
can
progress. It's still work in
exactly.
progress.
Yeah, for sure.
That's super cool. So now I wanna take a different question here. How did you find this interest in the first place? So you have a computer engineering degree from
Mm.
University of Toronto. How did you find yourself in computational biology, learning about single cell technology, learning about all these genomics research? How did you end up here?
Mm. Let's see. Good question. So actually, my path to this field it's very interesting. So both of my parents are doctors.
Wow.
So yeah, so I admire my parents from a very young age, to be honest. So like I feel like super cool. They just like go to the hospital. They cure patients. They help them to relieve uh their pain, something like that, right? So I found that super cool. And I always want to be like in the very young age. I think of being a doctor some days, but my parents say no because like being a doctor in China is very stressful,
Mm,
and also like some of the and also the total like the environments in China is not too good. Yeah,
right.
yeah, and so for example um like unhealthy work-life balance, and then very stressful for doctors. So okay, so because of that, my parents didn't want me to be a doctor in the first place,
Okay.
but I keep the seed in my in my mind. So like even though I go into the ECE, I try to explore all sort of opportunities of applying either like the software stuff into AI, into the healthcare, or the later the more fancy machine learning into healthcare. I
Mm,
remember in my second years, at that time, my goal is to create some kind of wearable medical device that everyone can use. For example, like to monitor something, like some signals in the body that
okay.
can give you some insights of the health situations, something like that. Like in my second years,
Mm,
I do go to a research lab. So that research lab is doing so it's wearing a cameras for capturing the hand motion functions, hand motor functions. So basically, can by capturing the hand functions videos, we can analysis how well the patient recovered from spinal cord injury.
oh, so we
Yeah,
can
so
track that progress of the recovery.
yeah, exactly. But
Wow.
yeah, that's the thoughts in my second year, and later on, I find that I don't find that too interesting to me. At
Right.
least that research experience to me is not too fancy. And
Right.
like by that time, I my original goal is create something like a product like apples that everyone can use, and I can be famous, something like that.
Yep.
Yeah, yeah. But
And health
but
applies to
it's
everyone.
exactly. So, but turned out to be like it seems pretty hard to create a universal medical device for everyone, right?
Mm-hmm.
So I kind of explore the other directions, which is combining with machine learning into healthcare data. So in my third year, I go to Wang's lab and Goldenberg's lab. So I did some research there. So Bo, it's one of the professor that have lots of foundations in the single cell data experience. So one of my summer research project is on single cell cancer genomic integrations. So by
Single
the end of
cell cancer? Sorry,
yeah,
which?
single cell cancer genomics data integrations.
Okay, right.
Yeah,
I
so
talked
like
about this.
yeah, so after that summer research, I I eventually did very well in that project. I created algorithms for doing very good data integrations for various type of single cell cancer genomics.
Mm,
And I find that okay, my work probably it's very useful for those cancer research based people, and then probably my work have the potential to help patients, to help a broader community, right? So I
mm.
feel um, a fulfillment in my mind, like in
Very,
my heart.
very altruistic of you.
Yeah, exactly. So like starting from there, I'm like, okay, I think this field is pretty good, and I'm interested in that, and also give me a fulfillment and satisfactory, right? So like I started to dive deeper into that field, and I look into other stuff.
Oh man, that's a great story. I really admire the fact that you found something that actually has like downstream impact on like people's lives.
Hmm.
I 100% like support that. But I also want to ask, there's so many different like aspects and fields and
Mm,
categories of healthcare and biology. There's like you could do like neuro, cardio, other like bodily
mm,
research into like curing so many diseases, born like genetic defects, like, and then there's actual matter of diagnosis, like how do you better diagnose these patients with like faster and more accurate. Then there's also the problem in healthcare systems, like like how do you make it
mm,
not as costly, surgeons, operations, billing, like it's so large and the impact is so huge.
that's
Like
true.
where how did you like choose like end up here, and like do you think there's other larger kind of ways you can make impact, positive impact downstream?
Yeah, that's very broad questions. So for healthcare data, there are many types, right? For example, as you mentioned, like for example, we have the MRI, the CT. So those are the image, right? And then we can apply um, like the very current computer visions technologies into those kind of data. And we also seeing NLP applying to healthcare. For example, like extracting informations from doctors' notes, doctors' prescriptions, and all sorts of like the healthcare informations, right?
Right.
And then I think Gary, you also have some experience with NLP in healthcare, right?
Yeah, it's a big mess of a field. There's
Yeah.
the current, at least from what I can, it's like by 2025, healthcare data alone will exceed
Mm,
all the data capacity, and it's blowing up, and we need a way to sort and organize this healthcare data. But that's just like one of the many problems that healthcare systems are facing.
mm. And the point is like there are so many different type of data, so many different questions in healthcare. So like for example, for NLP, I'm seeing people like using just NLP um, to summarize the like the latest medical papers from PubMed. So like for example, those papers are discovering a new molecules, discovering some new mechanism, but just like by human, we reading all the paper is too hard, right? So we want some tool to help us summarize the information. So something like that is also a healthcare questions, right? So it's like the whole field is very very broad.
Right. There's so many different options that
Exactly,
you can help with.
and also single cell genomics, as I mentioned for sure, is another very big field.
So
And
another very common question that people ask
mhm,
is AI going to replace doctors, or am I going to have in the future, or maybe it works together? I don't know, but do you see a future where I can open up my phone, take a picture or answer some questions, and then there's no real doctors and AI in the back, giving me a diagnosis. Do you think that's ever going to be the future?
Um, at least not in the near future, in my perspective. I would say doctor probably is the most hardest job to be replaced by AI. So
Yeah.
like
Okay.
that's yeah. So like the people just like won't trust AI doctor. Like
Mm,
people won't trust the black box algorithms that just like giving me a score and the results and the predicted results, right? So just like it's it's very hard to convince the patient to believe in that.
right.
But a real doctor, it just like um people tend to believe more into real doctors. So the current trend is like building AI tool that assisted doctors for diagnosis, um, for doing for example like improve the doctor's efficiency, something like that. So the current trend is on this. But
Improving the doctor's efficiency.
yes, but I wouldn't say that it's not possible for replacement in the future, but it depends on the problems. For
Right.
example, yeah, so just a simple diagnosis or some cure suggestions, I would say yeah, doctor might be possible. But for actually surgery, are you really trusting robots for doing surgery for
Oh
you?
man,
No.
not me.
Yeah,
I want
not
real
me.
hands on real hands
Exactly.
operating,
Exactly. So like
especially when it comes to like these maybe but I did see I think you have a very valid point here because there's a lot of these machine assisted surgeries where there's the doctor and then just the robot is taking in the doctor's movements and like
mm,
removing the shaking hands for instance, and that could be useful. I would trust that,
mm,
but not like
mm,
only machine. That would be scary. I would not trust that.
mm. Same here, same here. So even us not trusting that, like the like we can say it's the younger generation that um favor technology, right? We don't even trust that we can be operated by robots. How can those like other people,
Right.
right?
So I have another kind of broad question. Understand your kind of vision in this field. So, the biggest causes of death. Obviously, it could be anything in different countries, but at least one of the bigger illnesses is like cardiac, diabetes,
Mm,
um, you know, heart health in general. Other and cancer is obviously a huge one. Um, so the question is then, it seems like a lot of these diseases can be cured by, if I have better genes, right? If my genetic disposition, didn't expose me to having, heart failure or, diabetes or at least type 1, or,
mm,
and like for instance, if my child has autism, um,
mm,
and so on and so forth. And currently the method to try to cure cancer, for instance, is chemotherapy or um, some other methods that I'm not too sure about. So
mm,
let's say like in the future where there's like billions of dollars poured into pharmaceutical research, into drug development, and billions of dollars poured into vaccine development for pathogenic diseases, is that like in the future, like hypothetically speaking, is genetics and genomics editing research and this technology in general broadly, can it like replace all these
what?
other methods of dealing with illnesses in the future?
Hmm. That's that's very hard to be honest, or at least so okay. So like the pain point is so you can only associate a certain disease to some genes, right? So you know that that genes might be related to those kind of disease,
Right.
but you don't know removing that genes will result any other stuff, whether it will resolve any other stuff, or
Oh,
just like
like
adding, yeah,
I
or
might get
adding new
another
gene.
disease. I might get two other diseases after removing one.
Exactly, exactly. And also, it's so hard. We can do experiments on humans. Right
Mm.
now, it's like I remember last year there's like a gene editing baby born in China,
Oh my
like the
goodness.
like like the word first gene editing baby. like the real
Well, do you know
human.
what was edited?
Um, kind of forgot some kind of genetic disease.
Okay, so it was a sick, like it has some
Yeah,
genetic illness or
yeah,
condition, and it was edited to remove that.
yeah. So like that prov, I believe is removing two gene of that,
Wow,
and there's
born
tons
in
of ethical
2020.
questions. There are tons of ethical questions just rise up from like the world community to just like to criticize the prop, saying that you cannot do that. Like
But
it's
this
illegal.
is an open door that cannot be closed. Like surely,
Yeah.
like ethical questions, like whether this is good thing or bad thing. Um, the idea is that it'll do more good than harm. So
The
you
the
like
major
there's
cons
I think this technology
Mhm.
won't stop anytime soon.
Yeah, I agree on that point, but the major concern is how do we validate that after the editings, there's no side effects, right? And
Mm,
who and how would you prove that there's no side effects? So like the human life, so like for now, just like less than 100 years, right? You don't know, like probably it's okay in the first 5 years, but after 10 years, something wrong might happen, right? But we don't know. It's so hard to prove, so hard to validate, and yeah, so like
that is
to
true.
come
How do
to
you
coming
like you
up with
never
a
know
very
because you
yeah
can't
to come
experiment.
yeah exactly to coming up with a very robust methods, I would say it's still very far from now.
So how the real problem here is result verification after you do run your you have no way
Yeah,
to tell.
yeah.
So what do you do? Like do you like like the popular at least like the most widely used method is to test it on animal subjects like mice or maybe
Mm,
even pigs because their
mm,
physiology is very similar to ours.
mm.
But that can only prove to a certain point like we are not
Yeah,
mice.
yeah,
Our genetics are
exactly.
completely different or mostly different.
Mm,
So I guess that is the biggest barrier between us today and the realization of this technology until like I get
mm-hmm.
to like do you think I can get my genes edited before I die? Like is that like totally, like prediction, like you could say whatever answer comes to your mind, but like what's your personal thought on like can I get my genes edited? Can I get perfect eyesight? For instance, I don't know if
Um,
that's possible by the way.
I I think that's possible. I think that's
Oh.
possible. Yeah, I'm kind of optimistic. Um, if you say by the end of our life,
Mm.
yeah.
So in the next at
So
least 50, 60, 70 years, hopefully
yeah,
by the time we are edited, we can edit genes, we can actually stop at age 40,
yeah. I don't want to have back pain when I go when I'm 50, right? If I can just have no more back pain
No
when I go 50,
way.
I would just devote my life to research, right?
This is fascinating. This is very interesting. So this is super cool. I can't wait to hear any additional advancements about this. How has so like one last question? We'll talk about like these technical topics and about your profession. Obviously, we can't slip the conversation about COVID.
Mm,
How has COVID changed the research landscape and in at least genetics research, and if let's say you do single cell technology to do analyze single cell, like can you profile the RNA sequence of a virus, and is there ways, like is this single cell technology also effective on viruses, and like
yeah,
potentially curing them or eradicating them or rid of getting rid of them.
getting rid of the virus, you mean?
Yeah, like can we somehow not like gene
Mm.
edit or do something with like use single cell technology to beat coronavirus or
Okay, so
variants.
like for like if we want to use gene editing in solving the COVID problems, probably that's not too possible because like that methods is not mature yet.
Okay.
Like right now, most of the ways to try to prevent or just solve the COVID problem is drug repurposing and vaccine development.
Drug
So
repurposing.
yeah, so it's true that we apply single cell into the COVID virus.
Mm-hmm.
For example, like the COVID virus got mutated uh, many times. For example, the latest one is coming like got mutated in England or South Africa. I'm not sure. I kind of forget the places.
Mm-hmm.
Yeah, but like it is applying the single cell technology to identify, okay, so which part of the virus get mutated, and it is a different subtype of the virus than before. That's sort of stuff. It's all using single cell technology. So
Right. That makes sense.
yeah, so and but I wouldn't say it's possible to use gene editing on the virus right
Or
now.
us, what if
Yeah.
we gene edit on ourselves to, become immune? Is that possible? Or immune system is a completely different subset of like the physiological system?
Hmm. That might be possible, but only if we can know that what type of protein can inhibit the virus,
Mm.
and then we know what kind of gene can produce that kind of protein, which
Right.
is kind of hard, right? So
Right.
far, I don't think there is like a evidence or just like 100% proof on this one.
Oh, I see. Okay.
Yeah.
Well, we definitely, certainly live in interesting time, and partially thanks to you. There's Elon Musk putting computer chip electrodes inside our skull,
Mm,
measuring EEG, and brain waves to and using Bluetooth to kind of help our lives. There's CRISPR-Cas9 tech gene editing
mm-hmm.
technology. There's single cell technology. And then there's the whole coronavirus pandemic. So definitely an interesting, a few like recent years with, healthcare and biology, computational biology in general. So thank you for all this information.
No problem.
So now I want to kind of take a step back from the kind of technical aspects, the professional aspects. So,
Mm-hmm.
uh, Rex, what do you do, like outside of, when you do your research? You said you work on a startup. Can you tell us more about that? Like, what does a computational biologist do outside of computational biology, AI, and machine learning?
Um, okay, that more related to my PhD work, right? So I would say a PhD work consists of three parts. So of course you have the research part
Okay.
that you have to conduct lots of research projects. You're trying to produce a paper out of it, and also for junior PhD, there is some course that you need to take, and those course, I found it's actually very interesting. So like there are two types of course you can choose. So first one is just um, like going in the right direction. So you get to know very like many other fields.
Okay.
So for example, for last terms, I did some like computer human computer interaction stuff.
Mm.
So I took the course and also took the computer graphics course. At least I know, okay, what's the like the current trends of those two fields, or what kind of technology, what kind of tool created by those two fields. And you can also, by taking those courses, you can get to know whether you're interested in those other fields. My conclusion is to those fields, for me, no. So like, it's also, yeah, to
You'll learn it's
eliminate
not
some kind of
yours.
options. Yeah. And the other type is just like go deep into some of the areas that you're currently researching on. For
Mm-hmm.
example, for machine learning, right? You can, so the course I'm taking this term um, teach me lots of math and like the what happened behind the like the just like the calling the architecture, like calling the tool, like what actually happened behind. How would you um,
Oh, and the machine
train a
learning.
better, exactly. So and other than that, the third part I would say is some other. I categorize that into communications. So by communications, you can go to, for example, you can go to different talks. You can, for example, everything is going to virtual right now.
Yep.
So like the lots of the big conference right now, it's going virtual. So it's actually a very, to be honest, that is an advantage part of getting off the COVID pandemic, because it just like people don't need to like travel a long distance to a conference to listen to the talk. Now just they just open their computers, right? So like and also it's for free. So so the knowledge sharing now it's more faster and cheaper. And also with that, like because in the computer science community, everything is shared. For example, if you publish a paper, you need to have a code for that, right? Now
Right.
it's the common trend, not like 20 years ago. So with that, you can use the latest code to do all sorts of thing that you want to do, and that also gives us opportunity to build something that you want, right? So like for example, one of the side project I'm doing is applying NLP stuff for teaching young children to learn a foreign language, for example, an English.
Right.
Yeah, so like it basically is an app that teachers can give out can send out assignments on the app. The assignments are some oral English practice assignments, so the students can use their app to see how well they pronounce some kind of a word and whether they can do some basic simple conversations, and also they can take some took some classes from there, and like we believe that in the near future we might just have a virtual AI teachers that replace some of the like the current tutors.
Wow. So I
That
guess
is
the
more,
the AI teacher coming earlier than
yeah,
AI doctors.
that's more practical than AI doctors, right?
Right. I would
So
take a AI teacher. Although that's kind of freaky, it's also very scary to think that AIs will be teaching us, in the future.
yeah,
It's like acknowledging that AI is smarter, or maybe at least it's just knowledge.
yeah,
It's a fuzzy topic. We're still not there yet.
true, true, true. So like yeah, link back to your stuff. So like be able to do that, you also need to communicate with other people. You need to get some new information, new idea. You uh, keep track of the latest technology, latest tool, so that you can apply either to your own research or your some kind of other side projects.
That makes a lot of sense. How much time do you like? I understand you're a very busy person, and once again, thanks again for coming on the show. How do you split your time? Like, how much time is required? Like, obviously, learning all of this, domain expertise in computational biology requires you to learn computer science as well as biology, not half and half.
Mm.
So how do you manage your time, and like how do you like what's your strategy to learn things and be quick about it, and learn so deep about this particular subject?
I would say you need to be clear on what's your priority for now. So like let's say so you need to set the goal for yourself at least in the short term. For example, in 1 month, I want to learn something, and then you can prioritize the times a little bit more on that subject for this month. And after this month, you can rotate to other subjects. I would say you always need to have one thing that's like a main theme of your time. So like it's very hard to like balance two or three more thing at the same time because that can get distracted, right?
Right.
So always have one thing that just like a main focus. But that doesn't mean that the other stuff is not important or I would just spend significantly less time on other stuff. But just like you need to keep in mind that okay, I need to treat that as the most priority stuff in this week or something like that.
That makes a lot of
Yeah.
sense. That makes a lot of sense. So prioritize your work, have a theme, have a
Mm-hmm.
short-term goal. That makes a lot of sense. Well, Rex, it's that's very useful information. So I want to end it off with one last question here. So
Mm-hmm.
you're in your second semester, I believe, or first year in your PhD. Perhaps it's a little early to ask, but where do you see yourself? Like, what's next for you? Like, are you going to be after you do your PhD? Is like, do you see yourself doing more research and staying in this research community, or would you want to obviously like the kind of landscape of this is that you researchers find new ways to do things, engineers go and implement them, product managers make it into a product. Where do you see yourself like obviously within this computational biology realm and healthcare space, where do you see yourself landing in, that whole like spectrum of things?
Mhm. Yeah, it's true that it's a little bit early for me to be really answering these questions, but at least I will try to explore the direction that at least I should focus on my research work, right? At least I should build something for myself in one of the field in healthcare. And like in the like after graduates, I'm thinking so far as to I can go to academic
Okay.
academia, I mean, yeah, but my but my personal interest is still in um creating a company, a startup that can solve a real healthcare questions that never be solved by existing companies.
Wow, that's
Yeah,
that's inspiring. That is inspiring.
the
Can you give me a sneak peek of like what kind of problems is it computational biology and this similar line of research that you're
um
doing or?
my recent really recent interest is in drug development. For
Okay.
example, like probably one way just like to how we can apply machine learning into fast drug development and also more accurate target rate, something like that. But um, not too sure. So like there's lots of field that can be applied on, and not every topic or every question are well studied by AI in healthcare right now. So I'm just like keep exploring and looking to opportunities.
Well, the good news is that there's, endless challenges and problems and questions in
Mm-hmm.
healthcare and biology. So, there's definitely no shortage
Yeah.
of things to do. Well, Rex, it's been a genuine pleasure. Thank you for all the learnings, and it's so fascinating to hear about the research that you've been doing and all the progress that's been made over the last number of years in this space of computational biology, and I can't wait to see all the outcomes of this in our lifetime.
Mhm. No problem, Gary. It's also a pleasure for me to be able to share this stuff and also coming to the show. Thanks for the invitations.
Great, thank you, Rex.
Thank you.